Hello everyone, and welcome to today's live panel on addressing the antibody reproducibility crisis, a panel discussion with key scientific leaders. We are delighted to bring you this educational event presented by LabRoots and sponsored by Thermo Fisher Scientific. Let's first introduce our panelists. Aled Edwards is the CEO of Structural Genomics Consortium and on the faculty of the University of Toronto in Ontario, Canada. He is also a member of the International Working Group for Antibody Validation, IWGAV. Anita Bandrowski is the scientific lead at the Neuroscience Information Framework at the Center for Research in Biological Systems at the University of California at San Diego. She's also the founder and CEO of SciCrunch, and she is also an IWGAV member. Paul Wallace is a professor of oncology and the director of the Flow and Image Cytometry Facility in Roswell Park Cancer Center and Institute in Buffalo, New York. He's also an associate professor of pathology at the State University of New York in Buffalo. Matt Baker is the Director of Strategy and Partnering for Antibodies and Immunoassays at Thermo Fisher Scientific. And John Rogers is a Senior R&D Manager for mass spectrometry reagents in the protein and cell analysis business with Thermo Fisher Analytic. Myself, I'm the moderator for today. My name is Christoph Hergersberg, and I'm a Vice President for R&D for protein and cell analysis and for antibodies and immunoassays. All right, so let's get started right away. But before we begin, I would like to remind you that this is an interactive procedure. This is an interactive event. We encourage you to participate by submitting questions during this event. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation, or use the Q&A button to let us know that you're having a problem. Thank you. Looking forward to your input. All right, let's get started. Antibodies. Antibodies are some of the most frequently used tools in basic science research. However, the lack of reproducibility standards have resulted in millions of dollars being wasted by researchers on antibodies that either do not work, or they do not work specifically, or they do not recognize the specific target. As a reaction of that, recently, the International Working Group for Antibody Validation was founded and convened in order to formulate the best approaches for validating antibodies that are used in common research applications, and to provide guidelines that ensure antibody reproducibility. Today, we will discuss the antibody reproducibility issue, the antibody reproducibility crisis, and what standards can be implemented to address the issue. Let's begin our discussion. So, to the panel, can you share your perspective on the reproducibility problem and the importance of antibody validation? Please also share any personal experiences and thoughts on discussion groups such as the IWGAV or GBSI. Let's start with Dr. Edwards.
Well, thanks. Well, those of us of a certain age have known about the fact that antibody reagents have been of questionable characterization, and this is for a 30 or 40-year problem. And it's interesting that no one did anything about it, and in large part because it's a very difficult problem to solve, involving sort of the complexities of market forces and scientific incentives. It really came to the fore maybe about 5 or 6 years ago when pharmaceutical companies started to publish the results of their internal reproducing, as it were, of academic literature, where they found out that a large number of early discovery projects couldn't be reproduced by pharmaceutical scientists inside their companies. And all of a sudden, a problem that had lay lurking for 30 years came to the fore. So I think that the commitment by Thermo Fisher Scientific to try and help fix it is an incredibly positive thing to happen. Now, it's not going to be easy because the problem has been around for 30 years, but I think recently there's some, as it were, game-changing technologies that have made it possible to put quantitative metrics on the quality of an antibody, such as CRISPRs to create knockout cells and mass spectrometry. And now I think it's possible to create community standard quality criteria for antibody that I think that the world can adopt. And it's important for us to help them adopt that. And it's great that you guys really want to be part of that, in my opinion.
Thank you, Dr. Edwards. Dr. Bandrowski.
From our perspective in the research community, one of the things that really is, adding to this whole problem and the whole dilemma is that really researchers, even when they get great results, the way that they're publishing those great results doesn't really allow other people to actually reproduce those results. And one of the simplest things that we're not doing as a scientific research community, which I think needs to be addressed, and it was addressed a few years ago in the Vasilevsky paper, essentially showing that over half of the antibodies that are published can't be tracked down to the actual antibody that was used. And that is a problem in reproducibility that's actually really easy to fix, right? You just need to ask people, the researchers, at the time that they're publishing, to identify the reagents that they're using. And this is a simple thing, and this simple thing has actually led to a fairly large reproducibility project called the Resource Identification Initiative, which I also lead. Along with that, we have a large database of antibodies called the Antibody Registry, which then tracks all of those reagents, whether or not they're still at one manufacturer or another. But it tracks those reagents along their life cycle. And we are extraordinarily fortunate, actually, to have some support from the Thermo Fisher Scientific family, because they actually recognize that this key problem, in addition to other reproducibility problems, this, again, is highly fixable. It's addressable, and it's one of the things that we can really get a big win on. So I'm looking forward to continued work with Matt and others on this.
Thank you, Dr. Bandrowski. Dr. Vallis?
Yeah, I have to really agree with my colleagues here, Anita and Alid, that there definitely is an issue. And to Anita's point about the fact that it's very hard to reproduce data. I remember going back when I was in graduate school trying to reproduce a procedure that somebody had published and basically throwing up my arms that it just wasn't working. And today, that's continuing. We're in the process of trying to validate an antibody, TCL1, and I've certainly seen good data with it, but you go through the protocols and you go through the procedures, and you just sort of throw up your hands as to what's going wrong with this. Mm-hmm. So thinking about this really from a clinical perspective, which is what half of my responsibilities are at Roswell, it's been very, very difficult to in clinical flow cytometry to get good reproducible data from one laboratory to another. I think there's a lot of tests that are like this, but probably the one that comes to mind, or at least in the forefront of my mind, is the ZAP70 test. Now, this is a test that was published as being able to tell you if a CLL sample was going to be more aggressive, and it should probably be treated more aggressively than to just watch and wait. So very high value to the patient. But when it got implemented, it was implemented in different labs in a variety of different ways. And different reagents were used to lyse the red cells, different antibodies were used, different fluorochromes, different ways of gating, different strategies all the way through. So that the final result became that if you got a result from my lab, which I'm sure was the correct one, was one thing, and if you sent the exact same sample out to another lab, you got a completely different result. And so the doctors began to know that, and they began to lose faith in that as a particular assay. So I think as a field, where we have to go with this is really to a standardization, a harmonization of reagents, and a harmonization of analysis. So it's a very real problem, and I do think that there are solutions to it.
Thank you, Dr. Wallace, and thank you all for these introductory statements. My question to Matt is, we have an extensive portfolio of antibodies. How do we view this reproducibility crisis, and what are we doing to support the research community in solving the issue?
Well, as you alluded to, we are a major supplier, manufacturer of antibodies, and so our customers are key to us, researchers in the research community, and antibodies and their reproducibility, and even what I'd like to mention, specificity. Confirming specificity is core to what we want to accomplish. A lot of what we have been focused on at Thermo Fisher is really looking at these new tools that we alluded to, and I think we'll be able to talk about here in more detail today, to how we can add additional data so the research community can really look at the attributes of antibodies. We've also looked at, we have over 75,000 antibodies, and how do we deploy a strategy to go forward and systematically use a lot of these new tools to add data. But what we've really heard a lot from the research community, and really this goes back to some of our very early discussions, and one of the main reasons why we supported the IWGAV was to really kind of understand what is the most impactful pieces of data and information tests, techniques that the research community really wanted to see. And what we found a lot, the answer, which may seem like common sense, but also is reassuring, is that they really just want to see transparency. They want to see all of the experimentation, the relative cell lines, the relative conditions, as much transparency as possible, and maybe back to Anita's point about the methods and how things were exactly used. They just want to know, so when they have that antibody in their own hands, they can have the confidence that will work, and it helps them get to their answers much sooner. We are taking a systematic approach, going through our portfolio and trying to identify the most highly used antibodies, the antibodies that are used in publication the most as our first target. Obviously, you can't do everything overnight, but we are committed and have really formed extensive groups within our organization to specifically address not only antibody validation for new targets that we're developing, but also retrospectively throughout our entire collection. And we've to date already gone through several thousand antibodies that we've sent back around the rigor of some of the standards, such as the ones proposed by the IWGAV, and that I think we'll again talk about in more details today.
Thank you, Matt. Thank you also for framing our approach and also bringing together the viewpoint of the academic science and us as an industry partner, that we really have this view of this being a common problem for the community, not individual problem for individual researchers or one company. So that already sets us up for a successful approach, and you have outlined that in the beginning. I would like to be a little bit more specific about the validation, and I would like to ask Dr. Rogers, how are we validating antibodies, and for both specificity and for application validation?
Thank you. Yep. So we talked about past history and experiences. My experience in managing mass spectrometry, protein mass spectrometry labs, is in research. We often find proteins that are regulated in response to some condition, and when an investigator publishes a paper, the reviewers often ask for some orthogonal verification of the results that we're seeing. And they're typically asked to verify their mass spec results with a Western blot. And it's particularly infuriating to some of the mass spectrometrists because typically their experience is they have to order 3 antibodies commonly to get one that works, quite frankly, for their Western blots. And many times these antibodies are poorly characterized, and it's not even certain that the antibody is really recognizing the target or is appropriate in terms of its application. So maybe it's validated for flow, but not for Western blot. Maybe it never will work properly because it's the way the epitope is framed. So our approach is to use one of these tools, one of these pillars, which is immunoprecipitation with mass spectrometry. So we precipitate the native target, and then we identify and verify the target of the antibody. And with that, we also identify additional information like post-translational modifications, isoforms, variants of the protein, and interactors. And that provides a lot of insight into not only the quality of the antibody, the performance of the antibody, but also it gives some implications into the biology of what that antibody can detect, can inform us about, such as what interactions may be coming along, what interacting proteins come along with that protein. So that's given a kind of an overview of the approach that we're taking and my past experience with some of these frustrations of a biologist trying to better understand the tools.
Thank you, John. This is a very important aspect on looking at very modern technologies that are relying on antibodies and relying on the validation, and the researchers being frustrated by the absence of that validation. So to that point, Matt, are there other methods of validation that we are using, and what are these additional validation and testing methods?
So yeah, in addition to the IP mass spec, or I guess inclusive of the IP mass spec that John discussed in detail, we really have 4 pillars or 4 methods, if you will, that we're looking at in addition to our standard application-based validation. So again, as John mentioned, we've got IP mass spec, which we're really excited about because of that definitive quantitation that you can get, plus the known and unknown interactors. And then second, we have genetic modulation or genetic modification. And this is really using siRNA for knockdown and CRISPR for knockout. Obviously, this is a really powerful tool. People have been using siRNA for some time, but now with CRISPR, the ability to really completely knock out the protein of interest and use a variety of readouts, whether it be Western blot, immunofluorescence, or even flow cytometry, to really confirm that specificity at a protein level using a great null sample. And then we have our third area is independent antibody validation. And this is where you use 2 independent antibodies, preferably to 2 different epitopes, that you can stain or test against the exact same protein samples, cell samples that you've prepared together. And you can imagine you can even do 4, 5, 6 antibodies at the same time. And it gives you a really great data set that is pretty comprehensive. And as you can confirm those binding patterns, those staining patterns, that those antibodies are indeed specific. It also allows you to really kind of put those antibodies against each other, benchmark them against each other, so you can get an idea of even things like species cross-reactivity, cell line expression patterns, etc. So a lot of additional great inferred data there. So the fourth pillar we're referring to as biological verification. This one is more of sort of a catch-all for really a number of different techniques inside this advanced antibody validation process. The first one being cell treatment. We think this one is really important for looking specifically at post-translational modifications and looking at phenotypic output and to build further confirmation that the antibody is specific. Another technique is looking at relative expression. And so this is where you would use like a null lysate, something that is natively expressed as low levels or even not expressed, and then looking at a high expression level. So the classical examples here would be something that is transiently expressed, perhaps developmental markers that are expressed in specific developmental stages, or maybe even tissue specific or cell line specific protein expression profiles. The next one is neutralization. So you could also, in this one, say, maybe even stimulation. So your classical agonist and antagonist. We really like this one because of the fact that it's not only specific for the protein, but it shows specificity for that active site of that protein. And then there's peptide arrays. So this one in itself, I don't believe is as valuable as when you combine it with a cell treatment. It's very important because obviously when you stimulate a cell, you stimulate many different things. And in order to really look at that specific motif, for example, with histone marks and dimethyl versus trimethyl, it's a great way to look at all of those individual peptides and know that your antibody is specific for that individual post-translational modification. And then the final one is or what we're more loosely orthogonal methods. Now, this was a separate pillar in IWGAV. We're kind of putting it under this bucket because we think there's multiple extensions and ways that you can apply this. And one of the key ways that I'll illustrate very quickly that we're interested in applying this is looking at Western blotting and correlating that with IP mass spec data. So you have the ability to look at the expression profiles and what you pull down and then to verify that then independently in Western blotting. So biological verification, again, a number of different techniques in that one, but I think another very impactful protocol. So in the end of the day, I think as we've looked through all these methods, nothing is absolute. But what we want to do, and again, what the community has really shown us or told us, is that they want more assurance that the antibody binds what it's binding, you say it's binding, and that it's going to be reproducible and binding for independent labs. And so we're trying to produce antibodies with more data, putting that additional data on the website. We're also trying to provide as many details as possible about the experimental conditions, which I think is also really important. So as you see us evolve over time, you're going to see more and more data. We hope we don't get to the point of data overkill, but we think to this point, more data is better. And for the researcher to be able to look at that data and have confidence and even be able to see how they can apply that data to their system.
Can I comment on one thing, Matt? I mean, when you said nothing is absolute, I would take issue with that. Certainly in the mass spec applications, it's a very high, statistically significant result when you get the peptide from your protein or a series of peptides from your protein in your immunoprecipitate. You can't fake that. So there are methods that are absolute. When you make a CRISPR knockout, if it's, the genetics are right, it is absolute. That protein's not there. So there are a number of techniques that are really higher value in quotation marks, in my opinion, because they're far more quantitative and less subject to the vagaries of the experiment. And you guys are championing that, so you should be, shout out to you guys for doing that.
So thank you. That is a great discussion, and thank you, Matt, for comprehensively stating all the efforts that we're doing. And then Al, thank you for commenting on the specificity of the methods depending on what people want to do. And that was exactly what I wanted to start now discussing a little bit more with the panel at depth. Now, when you look at it as a parallel, coming back to the original questions, we have to start somewhere. We have started somewhere. What is your commentary on what John and Matt have provided?
Yeah, okay. So I think that reproducibility is an interesting thing because there's no use reproducing experiments beautifully if it was a bad antibody to begin with. And so simply reproducing experiments is not the aim. The aim is to make sure that the antibody has quantitative and standard operating procedure kind of characterization. And that is best done, as Matt was saying, with genetically cleanest system you can, and CRISPR knockout's the best, and honestly mass spectrometry, where it feeds you quantitative results. Western blots are not quantitative, so that is always going to be user dependent. And so I think that you guys are doing the exact right thing in picking technologies that will give you as close to black-white as possible, condition-dependent. And I would highly encourage you to continue along those lines, because those results can't be gamed, right? They are going to be put into the public domain, I assume, the antibody validation by mass spec, the peptide counts, or whatever method you're using. I think that'll be a tremendous sort of flag that you plant in the community as to what is a proper validation of an antibody. So I would go, stay on course, as it were, for that strategy.
All right.
I definitely agree with that. I mean, and the other thing that Matt said that I really like is releasing more data. Because if the researcher can be more comfortable by seeing a whole pile of data as opposed to seeing one little, band in a Western blot that's been kind of cut out of a much larger picture, there's a different value that can be put on that, even if, whatever piece of data that, and I mean, first of all, it's wonderful to have the resources to be able to go after these very high value techniques, these very quantitative techniques, but even releasing the data about the Western blots. And I agree with you, they're not perfect, they're far from it. But in a lot of laboratories, that's what you see, so you want to see the comparison. So if you can see the comparison between here's what the company did, here's what I'm doing, and there's something wrong. So being able to just see that, being able to have that full data, I think is an incredibly valuable resource for the researchers.
Right. Yeah, if I could comment, it was striking. So a year and a half, 2 years ago, I was asked to try to figure out a way to validate antibodies using mass spectrometry. And we met with Alid and a few others and talked about approaches. And what struck us was the IWGAV paper, those guidelines are general guidelines and don't provide specific details of here's how you should do it, which I think is great, because at this point, this is a research effort. We're trying to get our arms around what's the best way to do this.
I think to the overall discussion, if we're talking about validated reagents with protocols using CRISPR cell lines or different methodologies, it would be nice to have coming out of the companies different, very specific protocols that if I purchased this particular antibody or I was to use this particular reagent and I followed this procedure, that I could actually get that result. I mean, there's certainly a lot of variation from one laboratory to another, but if I had a straightforward protocol that we could follow that worked with a particular reagent, then I would feel comfortable making modifications to it, my procedure, to see if I could improve on it. But at least I would have a quote gold standard that I could use. So I'd like to extend the discussion into flow cytometry, because I think that's probably an important part of the portfolio.
Yeah. If I can just add to that a little bit, flow cytometry is great.
Thank you.
But one of
Thought we'd
but
finish right there.
the somewhat less scalable moment is CRISPR and genetic validation, right? And the reason that genetic validation is so important and yet less scalable is because we really actually have a lot of techniques where different portions of the epitope will be present in the particular tissue. So I've done a little bit of immunohistochemistry, where you're in a full tissue. It's not the protein anymore. It's not the naked protein. It's not the raw protein. So you're not going to have accessibility to a lot of those parts, a lot of those epitopes. So a lot of antibodies are just not going to work in immunohistochemistry, where they do work in Western blot or another protocol. So one of the IWGAV pillars is that you need to actually validate for each individual thing. So yes, the antibody must work. I think that is something that the company has to show. And I think absolutely, positively, I totally agree with all of you guys on that. But in a particular application, it still might not work because just the epitope is not accessible in that particular application with that fixation and everything else. So really, one of the things that I would love to see out of some of the big public projects like COMP would be an accessibility to all of the data that they're actually generating, knocking out all the different genes across the mouse genome. And I would love to see some of the tissues from that become the standard, actually, because we've been trying to kind of get them interested in this, but this is just something that they're going to have to kind of look at. Wouldn't it be great to actually also have a standard for immunohistochemistry? And I think we have a lot of the pieces in the community where we would really be able to address that, but they're not yet coming together properly. But I think they will. I think over the next few years, as more people look at this, as more people understand that reproducibility is a very big issue, it's a very significant issue, I think these are the kinds of things that will be starting to come together.
Yeah, I certainly agree. There's a lot of antibodies out there that will work really well with in a Western blot that completely fail when it comes to immunohistochemistry, and likewise, there's a ton of antibodies that work well by flow but don't work in immunohistochemistry or don't work when you do a particular technique. So
Yeah.
I do think that when you begin to think about how do you go about validating, it isn't just in one methodology or one technique. It really is a variety of techniques. And I think that really is the issue. Is that you'll see an antibody go, oh, this recognizes this epitope, I'm going to use it, and then as you get further and further into it, you realize, well, it works in a Western, but it doesn't work in another technique.
I mean, it's important not to be nihilistic, but for immunohistochemistry, when we say it has been validated, unpack that. How did we do that? In the absence of knockouts, in the absence of this, it stains well, it doesn't stain well. I think that we need to be kind of careful, and so, for example, a flow that a set of antibodies are proven quantitatively and statistically to immunoprecipitate the antigen. So, at least this set of antibodies binds the protein.
Yes.
And then take that subset and screen them against the mouse knockouts
Yep.
or the human knockouts if we exist. That seems to me a logic flow, because at least you're starting from a point where that antibody is proven
Yeah.
to bind the, as opposed to screening de novo on tissue and you see some staining, a little more, a little less, stain a little more, and you get a different color. So I think it is I think we really got to go back to what you guys are doing at the very beginning and use some quantitative, statistically significant, and by quantitative, it doesn't mean I do doseometry and say, oh yeah, I get a stronger signal. Linear, titrate, all the important stuff that old biochemists used to do. These are biochemical reagents, as it were, and we should not forget that.
Yeah, I think that's a great point. So, again, the way that we look at things, and maybe this is where we need to turn things on its head a little bit, is we first go through all the application screening. So, to your point, we do all the flow cytometry testing, and we try to, pick out the correct models and do everything. And, that's how we, in essence, validate and say that this is, tested and confirmed for this application. But doing one of these additional tests first, is sort of the new thing, right? So, Al, I think you made the point before, why don't you screen everything by IP mass spec and then screen all the applications, right? And using it as a filter, if you will. There's still, some, differential display of epitopes and how those epitopes bind and those things. But the way that we're looking at this is, to your point about flow and flow being a pillar, is we're not looking at the actual application being the pillar, it's the technique to prove the specificity. And so, for example, how would one of these be most useful inside of a flow application, right? Is it coming up with potential knockouts, or is it having the correct cell lines, the endogenous, cell lines with absence or presence? I mean, what is that, standard of applying really your best case controls to prove that an antibody is specific in flow cytometry?
Yeah, I think while I would consider flow a technique, I think really there's a lot of, your point is that there's a lot of techniques within flow that can be used, and I think that really is where it falls apart. Is that as I mentioned, one person's technique is completely different than another person's technique. So that actually having as part of that validation or part of that the protocol that was used, that you could then go back and actually follow would make a lot of sense.
Yeah.
And that is really a great point on how we approach that overall. And what I liked about what you said was both procedures, giving procedures for specific applications, so give clarity to the researchers, maybe up to the point where we will say, don't use this antibody for that.
Yeah,
Won't work.
we've talked about negative data. We've talked about doing
Negative
that as
data,
well.
really,
Yeah.
because it also adds to the trust part of it. And then I wanted to ask you, Paul, on flow cytometry, because the immunology community within flow cytometry has really started a lot of this work back in the day with the cluster of differentiation workshops. It was a workshop approach, and people came with their data and they discussed them. So my question is, when you look at this approach that has given us some 350 CD labeled and agreed upon by the scientific community antibodies, or at least antibody specificities, what can we learn from that approach for the broader aspects of antibodies, and how is the cytometry community going further with that?
Yeah, I mean, certainly that approach made a lot of sense, where you would send off to an investigator your antibodies, and you'd say, well, I think this is for CD64, and they had a cell line that expressed CD64, had been transfected with CD64, another one that didn't, and you could compare the results. So to me, that approach made a lot of sense. I don't think the organization has really continued on with that. We sort of stagnated at whatever it is, 352 different CDs, and it sort of left at that. So I do think that approach made a lot of sense. It's certainly something that an initiative could be started up again to try and do that. So I think, to answer your question, that whole HLA approach made a lot of sense. The scientific community was more than happy to participate in it. There were publications that came out of it, and there was a lot of information just in terms of what those different reagents reacted with. My example, CD64, it was not only on monocytes, but low levels on granulocytes and that type of thing. So I think that was very informative.
So thank you, Paul. That is really very important and very instructive. Can you elaborate just a little bit more on what is the concept of the CD workshops, and how can we apply it for future approaches in having a community-based approach to validate antibodies?
Sure, absolutely. So the concept of the CD workshops was basically where an individual was who was really known as an authority in about a particular antigen or in that particular field. And I love the example of CD64, so I'll use that. Where an individual who really knew CD64 had a variety of different cell lines that were associated with that expressed this particular antigen, would then receive antibodies from a lot of different investigators or from industry, who would then he would be asked to screen these different antibodies against his cell lines to see if they were expressed by those. And there were positive controls, there were negative controls. What came out of that was a very clear delineation of the reagents that were actually recognizing that particular epitope. So you knew this particular clone, 10.1 or 22.2 or 32.2, that recognized the CD64 molecule. You would also know if there was cross-reactivity. They'd often look at cross-reactivity between the reagents, and as I mentioned, you would usually end up with a publication of it in a book or chapter, but a publication that really described that particular antigen, so that I know as a researcher, if I was looking for more information on it, the CD books were really very valuable just to go back to, just in terms of being able to find out what was the general reactivity to it. So I think in terms of the field, that was really a fundamental step forward, and it was also then really applied to some of the interleukins as well. The issues though, are with the CD64, or with, I guess a good example of this would be CD59, which is associated with CD59 is on red cells, and it's on a variety of cells, but it inhibits complement activation. And it's very important in the diagnosis of paroxysmal nocturnal hemoglobinuria. Well, there are a variety of reagents that we use in flow for this. Or a variety of reagents that are available for users in flow for this, but there's a huge amount of variation. So they all recognize CD59, but it's going to depend on the floor, it's going to depend on the clone, it's going to depend on the reagent. So that beyond the CDs, there's a whole nother step that needs to go on, just in terms of being able to get, really identify which clones, which conjugates, which forms are really going to be better than another. And I've certainly seen this all over the place. So yes, I think the CDs have been a huge help, are a huge help to the researchers. I'd love to see it move forward. And I think that there's when we talk about a community effort, that's exactly the type of thing that we're talking about. But in addition, I'd like to see where we really began to look at all the different reagents that were out there and began to identify ones that really were outstanding.
Best in class.
And that's a great statement that brings me to the question, are there questions from the audience? Because we've talked a lot about the different methods, but more we talked about the applications that are used by the customers. So for you are visiting this webinar, and thank you very much for that. Let's open for a few questions from the audience. The first question I would like Anita to answer. How do you see grants and publications addressing the reproducibility issue in the next 5 to 10 years?
So I'm really glad you said 5 to 10 years, because I think a lot of these kind of initiatives and things that have to change are going to take time to kind of go into the community and really work their way through. So we know that in 2016, I'm sure you're aware, in 2016, the grant criteria for review has actually changed. It's been updated. It's moving towards rigor. But this is going to, again, take some time to kind of get going, to understand and to change the way that standards, that the standard practice is happening. So I think over the next 5 to 10 years, I think we're going to see much more review of these grants moving towards the authentication of biological reagents, again, specifically antibodies, and also cell lines and other things. But we're going to start to see more people paying attention to rigor. And I think this is a really important thing, because, again, this is something that the whole community can take. And I would hope that some of the other funders would follow suit with the NIH, but right now, NIH is, although it is the 500-pound gorilla in the room, I think it is just one funding agency, and I would hope that other funding agencies would come along. So it's interesting with journals, and how do we actually work with journals? So journals over the next, again, 5 to 10 years are going to, again, be bringing elements of rigor and aligning themselves with the National Institutes of Health. And they're already starting to do that. So if we look at Cell Press, they have done a tremendous job moving all of their journals, again, slowly starting in 2016, towards reproducibility. And the first thing that we're tackling, and this is only step one in a many-step process, the first thing we're tackling is identification of those reagents, because this is a very easy thing to do. It's a step that we can all take together. And then once we take that step, there will be other steps. We will, again, align the papers with the grant review guidelines towards rigor. And I think this is, again, it's going to be a multi-year process to just get the community sort of on board. And then we can take these steps. If we take them together, I think they will be very, very powerful. So these are things that every researcher can actually do today. They can start to look up the RRIDs, so this is the Research Resource Identifiers, for all of their antibodies. And, hopefully, if they have some great data that they can link out to from Thermo Fisher Scientific, that would be, those links are available. So really, I love the fact that you said this is going to be a multi-year process, because it is absolutely going to be a multi-year process. And as a community, we're moving towards rigor.
Thank you. And Al, maybe you want to add to that, since you spoke strongly about standards and the community effort?
Yeah, I think I like the idea of tackling the funding and the publication, because there are a million biomedical researchers out there. Getting behavior to change is going to be difficult. But I think I heard it first with Francis Collins said, it's impossible to herd the cats, but you can move their food. And so, you know, there are two funnels to the scientific process that have far fewer actors, and that's where we get our money and where we publish our papers. So that's what we need to influence. But even if we try to influence them now, without community standards, there's no way to be able to say, is that a good reagent or bad? And the validation is in the eyes of the beholder. So I think the sort of one of the highest priorities is for the community, including industry, to get together and to define what a target product profile is, as it were, to use the industry term, of what would a validated reagent be and what are the minimum criteria that they needed, and then have the funders and the journals start to accept that. In the absence of standards, it's not going to work.
Thank you. Thank you both. Thank you for that question. I hope it was answered correctly. And we have another question coming in that I would like Matt to answer. How do I know if the antibody that I'm purchasing has gone through a validation process?
Well, I think that maybe goes back to a little bit of what I was referring to earlier, in that we really kind of see this as two distinct sets of criteria, right? So we've always done extensive applications validation, right? Western blotting, immunofluorescence, flow cytometry, to confirm the antibody works in the application. We see that as the absolute minimum threshold, and that's again where we're adding even more detail on the protocols that we're using, more and more data on different cell lines, different species, reactivity that matches the data sheet. So our goal there is to really have a piece of data that represents every single thing that is shown on the data sheet. Where the reproducibility in these, what we're loosely calling advanced validation or specificity testing. So what we're doing there is we've come up with a badge to represent antibodies that have gone through the additional testing. And that badge will be highly visible on the search results. When you're looking for an antibody, there's a list of antibodies on the site, and you'll be able to clearly show those. Now, what's important to remember is that, we've been doing this for a little over a year now. And as I mentioned, we've gotten through a couple thousand antibodies. We're going to continue to do this and add data over time. So when you look at that, you see a badge, and that badge, by the way, is also very present on the what we call the product data sheet, along with the specific information where we show the IP mass spec data or the knockout data, the siRNA data. But if you don't see a badge, that doesn't mean that it hasn't been qualified. It's just, we may have not simply got to test it yet. But through this process as well, something that I've talked with various members of the community is that we are taking antibodies off the market. We have some antibodies. Thankfully, we haven't had to do that a lot, but we have had some antibodies where we clearly show, either through mass spec or through various methods, that it's either cross-reactive with something, or we just simply believe that this additional data is worth removing the antibody. We also have scenarios where it's giving us more insight into the type of application. So we find that it may be validated for a couple of different applications, but this additional data, this additional layer of data, is providing us more guidance and confidence towards the full data set of applications that we're able to recommend and guarantee to the customer that the antibody will perform under.
Thank you, Matt. There is one last question, and thank you for the question. There is one last question that I wanted to ask Paul, and that regards flow antibodies. We have talked quite a bit about flow antibodies and flow conjugates, and Paul has explained to us that it's really the experimental system that is important there, the system of the reagents and the instruments. So I'm not surprised that he's getting a question on what would you do to select a flow antibody, and what data would you like to see?
So how would I select a flow antibody? It usually would start off with a question. What is the specific cell that I'm interested in. It could be a dendritic cell. It could be that the investigator is interested in an MDSC, or probably the most recent one for me was these monocytes that are going through a translational from classical to non-classical. And so I have a question, and then the next piece is, well, what are the antibodies that I'm going to need to address that specific question? So typically at that point, it's going to the websites, going to each of the manufacturers and looking to see what's available, what clones are out there, what the fluorochromes are, and I begin to think about putting that panel together. In terms of picking the clone, if I saw something like you're describing here, Matt, where there was a seal that said that this had been validated, that we knew that this was going to work well in flow cytometry, and I saw a protocol that said this is how to use it, then I would that would be something that would definitely attract me to that particular reagent. Alternatively, I might call Anita up and ask her what she's found that has worked in her hands, because she's the expert in something along that line. So it's basically a process of selecting the clones. Then I begin to think about what the fluorochromes are going to be, what's going to happen in terms of crosstalk between one reagent and another reagent, depending on how highly it's expressed. So that would be the initial process. I would bring those in. The first thing that we would do is titer the antibodies so that we know that we're working with a saturating concentration that isn't at a high level that we're seeing increases in background. So our first step is really to titrate it. Then we would basically validate the panel. We'd put that panel together. We'd run a number of controls, which in flow we call them FMOs or fluorescence -1s, but we would basically validate that the panel itself is working well. And lo and behold, it probably won't. And so what that really means is that I've got to go back and reconfigure the fluorochromes so that I'm getting the best separation in all the populations that we've got. And that's typically the process that we go through. To speed it up, it would be great if we had protocols, we had examples of where this particular antibody worked. It'd be even better if the vendors provided panels that they had pre-validated that we could then go through. So that's basically our process.
Thank you. Thank you very much. And I think your summary also shows that flow cytometry in particular, based on the complexity of both the applications and the technique itself, has already established over the years a process for quality validation that has been accepted by the community. So I think we can look forward to learning from flow cytometry on how to more broadly approach that, as we are including the community and the funding agencies and the journals into that process. So with that, I would like to once again thank our panel of experts. And I'm very pleased that Thermo Fisher Scientific was able to make today's educational event possible. Now, before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through May of 2018. And you will receive an email from LabRoots letting you know when this webcast will be available for replay. And please share that announcement with your colleagues who may have missed today's live event. This concludes our discussion. Thank you for joining us, and stay tuned for the next event. Thank you.